Okay folks, so this is the podcasted version of the DNA transcription lecture. So the lecture outline, the overview lecture, is we're going to talk again about transcription briefly, just to introduce the topic, before going on and looking at the structure and function of RNA polymerase II. Initially by looking at the RNA polymerase II core promoter, and then how the transcription complex, this preinitiation complex, actually forms at this promoter. A major component of that preinitiation complex is the RNA polymerase II, and we'll talk about that in addition to some of the other preinitiation factors, before discussing some of the special features of RNA polymerase II, which make it an extremely clever enzyme with regards to regulating transcription. We're going to mention then a complex which is unique to eukaryotes called the mediator complex. So one of the reasons why transcription and transcriptional regulation is far more complicated in eukaryotes than it is in prokaryotes. We'll talk briefly about what that is. Before mentioning proofreading during transcription, so I mentioned in the DNA replication lecture that some of the DNA polymerases possess inherent proofreading capabilities. The same is true of transcription. There are inherent proofreading mechanisms that operate during transcription as well, to make sure that the DNA is correctly read, so you transcribe the gene accurately. Talk then about how transcription is terminated, so what happens when you get to the end of a gene, how does this big complex disassemble, before finishing up with a few discussions around the regulation of transcription. So how is one gene expressed at a higher level than another, how a gene's repressed in certain cells, whereas expressed in others, that sort of thing. And finally, like we did in the DNA replication lecture, we'll also then link errors in transcription and the transcriptional machinery to the development of specific diseases, before giving you a summary slide just to bring everything back together. So the learning outcomes then are that you need to be able to describe the three eukaryotic RNA polymerases and their associated functions. So as we'll see, there are three polymerases in eukaryotes and humans. You need to know what those three are and what they do. You need to understand the structure of this so-called core promoter, this RNA polymerase II core promoter. And then importantly, you need to be able to discuss how the preinitiation complex forms on this promoter. So the process, the order that it occurs, etc., that sort of thing. You need to give a pretty good explanation of what transcriptional elongation is, importantly how it's actually initiated, what factors control its initiation, and also then discuss proofreading and termination of transcription, that sort of thing. The regulation of transcription by these DNA binding proteins is something else that you need to take away from this lecture. So if once you leave the lecture, you might want to do a little bit more reading about that, how these DNA binding proteins actually go about regulating transcriptional levels in the cell. And you need to be able to give some examples of diseases which are associated with errors in transcription and the transcriptional machinery. So we've already discussed in the replication lecture, and I'm sure Dr. Porter touched upon this as well in his lectures, that DNA is a molecule that harbours a genetic code. So in the replication lecture on Wednesday, we looked at how this information is passed on through generations. What we're looking at in this lecture is how that information is actually used by the cell. In other words, how it's expressed. And in some ways, you can consider that replication and transcription are similar. Some of the basic tenets of replication also hold true for transcription as well. But there are a number of differences, a number of significant differences in how the mechanisms work. Not least, obviously, the proteins that are involved and the mechanisms that control those proteins, or the mechanisms of which those proteins form a part. So you can see here that one of the things, obviously, which is similar to DNA replication, is that you need to melt the double-stranded template so that RNA polymerase II can start copying from one of the single-stranded regions of the gene. And what happens is RNA polymerase II moves along the gene and starts producing this RNA, which emerges from the gene as the polymerase moves along the length of the DNA. Now, one thing to realise, and this is important, and it's something to bear in mind with everything really, is when you start imagining how these things work in your head, often it might not be the same as what actually goes on in real life. So you might think then that a polymerase comes along, certain factors kickstart a transcription, and the polymerase moves along the gene, copying an RNA, and then at the end of the process, a piece of RNA falls off, and that's what goes out into the cytoplasm to be made into protein. In reality, it doesn't work like that. Again, it'd be a very inefficient way of the cell working if it was just to wait for one polymerase to move down the length of the gene to produce one RNA. So what actually happens during transcription is once gene expression is turned on, the polymerase will start copying the gene sequence and producing an RNA, and then more or less as soon as one polymerase has left the promoter region, another one will bind and follow suit. So it's kind of like the M62 at 5 o'clock, it's just nose to tail with polymerases along the length of the gene. And what that causes then, if you look at electron micrographs of genes that have been expressed, is this so-called Christmas tree structure. So if you look at the electron micrograph here, you can see that what we have is lots and lots of, if you like, little strings, little arms coming off of this long gene here, and each of those little arms coming off represent an RNA that's been produced by RNA polymerase. So you get, what this allows the cell to do in response to a signal is to very rapidly from one gene actually produce hundreds, thousands of RNAs at high speed, which then can get translated in the cytoplasm into protein. So, one of the things that you need if you're a multicellular organism is very rapid response to external cues, and this is one of the mechanisms which allow mammalian cells and multicellular organisms to function in that way. So DNA makes RNA. This is what this lecture is all about really. I've already said it's more complex than the situation in bacteria. One of the reasons, the most basic reason it's more complex, is we have 3 different polymerases in eukaryotes. There's RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. 99% of this lecture is going to be about RNA polymerase 2. RNA polymerase 2 is the RNA polymerase that's responsible for making messenger RNA. So that's the polymerase which makes the RNA that goes on to be protein. We'll talk briefly about what the other 2 do at the end of the lecture. But for the majority of the time we're going to be discussing this, it's going to be specifically referring to RNA polymerase 2. So RNA polymerase 2 is a really gigantic molecular machine. It's the best way of thinking about it. It's about half a megadalton. So it's a huge multi-domain, multi-protein complex. But even given all this complexity and the size of the protein, it's not capable of actually initiating RNA synthesis by itself. It needs associated factors in order to be able to do that. So what are these associated factors? Well, they're known as the general transcription factors, or GTFs, and together with RNA polymerase 2, they associate the promoter to form what's called the preinitiation complex. And until this preinitiation complex is formed at the promoter, you don't get any gene expression. If you break it down to basics, it's a very similar process. It's analogous to the situation that exists in E. coli. I'm sure you've had lots of transcription lectures about bacterial genes, etc. And this is where basically the sigma factor, so-called sigma factor, is required in order to recruit the RNA polymerase in bacteria to kickstart gene expression. We don't just have one factor in eukaryotic cells, we have many, many, many factors, as we'll see. So what do eukaryotic promoters look like then? So I've already mentioned the fact that RNA pol2 requires a number of these general transcription factors in order to work. This is in vitro, so this is if you try and do transcription in a test tube. You can't just put RNA polymerase 2 in, some ribonucleotides and some DNA sequence with a promoter. It won't work. You also need to supplement it with lots of these general transcription factors. Situation in vivo in the cell is actually even more complex. So you can kind of get away with a minimal selection of transcription factors in the test tube. There are more factors required in real life, if you like, to kickstart and make transcription work in the cell. And that's because of a number of reasons that we're going to discuss during the next few slides when we look at preinitiation complex. But basically it revolves around the need of these different transcription factors to bind at the promoter sequences in order for transcription to start. So this is an example, if you like, of a canonical or just a bog standard RNA polymerase 2 core promoter sequence. And some of the sequences will be familiar to you, you'll have heard them mentioned before, and some of the similarities, there are similarities that exist between the bacterial promoters and the eukaryotic promoters as well. So the really important elements are obviously the Tata box here at -25. So -25 refers to 25 bases before the start of the gene sequence, the actual coding region of the gene. The Tata box is bound by a special protein called Tata binding protein that we'll talk about in a second. Then there are also these other key elements. The initiator element is a special element that exists in nearly every core RNA polymerase II promoter, and this is bound by TF2D, one of the transcription factor associated germinal transcription factors that form part of the preinitiation complex. And then sometimes you can have downstream core promoter elements, so sometimes you have specific sequences which actually reside within the gene itself, which associate with the preinitiation complex, and also you can have recognition elements upstream of the Tata box, which bind to different components of the preinitiation complex. In this case, this binds to the transcription factor 2B, TF2B. Now, as we'll see at the end of the talk, in humans and in eukaryotic promoters, there are also regulatory sites that are very distal to the gene itself, sometimes thousands and thousands and thousands of base pairs away. Now, that phenomenon is not unique to eukaryotes, it exists in prokaryotes as well, but it's a lot more common in eukaryotic promoters and eukaryotic genes for them to be regulated by these very distant elements in the DNA. And we'll talk about examples of that and how these actually work later on in the lecture. But in essence, if you break it down to bring it down to brass tacks, these transcription factors and RNA polymerase II, effectively they're functioning in a reasonably similar way to what happens in E. coli. All the germinal transcription factors can be thought of as a sigma factor, and then RNA polymerase II is basically the RNA polymerase from E. coli. So in other words, you need all these germinal transcription factors to bring in the polymerase and then kickstart gene expression transcription. So what actually happens then during this pre-initiation complex formation? So the first thing to realise is it happens in a specific order. So they don't just all mill around a promoter and fall onto it randomly. There's a specific order of recruitment that goes on, so that it's a regulated process. So the first thing that actually binds to the promoter is the TF2D complex. So this is a multi-protein complex, a huge multi-protein complex in its own right. One component of which is this Tata binding protein, TBP. And this protein recognises and binds to the Tata box region of the promoter, and then facilitates the recruitment of other components of TF2D. Now, Tata binding protein does one very important thing when it binds to the promoter regions of genes. Because it uses a rather unusual beta sheet mechanism for interacting with the minor groove of the DNA molecule, as opposed to most other DNA binding proteins, which actually use alpha helices and interact with a major groove of DNA. What Tata binding protein does is it changes the structure of the DNA. So this beta sheet interaction, which Tata binding protein does with the minor groove of the DNA, causes the DNA to actually bend, and it bends quite significantly up to 80 degrees, 80-90 degrees, at the point where Tata binding protein binds to. And this is thought to be important then for recruiting in the rest of the pre-initiation complex and kickstarting transcription. So after TF2D binds to the promoter, we then see recruitment of a second transcription factor called TF2A. And what TF2A does is it effectively stabilises the binding of Tata binding protein to the Tata box. So the Tata binding protein gets recruited to the Tata box, and then TF2A is brought in, and that kind of stabilises and locks the entire thing down. Following that, we get binding of TF2B. So this binds asymmetrically to Tata binding protein and forms a bridge between TF2D and RNA polymerase II. So following binding of TF2B, we then get recruitment of RNA polymerase, which comes in with another transcription factor called TF2F. So these things arrive together. TF2F and RNA polymerase II arrive at the promoter together, and they bind this Tata binding protein TF2B complex, which is formed at the promoter, for stabilising it even further. Then the final parts of the pre-initiation complex to arrive are TF2E and TF2H. And TF2H in particular is interesting because it's another really huge multifunctional multi-protein complex in its own right. And one of the roles of TF2H is to actually melt the DNA, so to give access to the single-stranded sequence for RNA polymerase to copy the gene sequence, and also to facilitate elongation, which is the first step of transcription once the actual polymerase leaves the promoter and starts transcribing the gene. So I mentioned earlier that RNA polymerase II is an interesting enzyme. It's got an interesting structure. One of the most interesting aspects of RNA polymerase II in eukaryotes is the so-called carboxy terminal domain, CTD. It's also known as the tail. And this carboxy terminal domain, this RNA pol2 tails, really a site which can be highly modified by post translation modification by phosphorylation, etc. And it's thought to be the major regulatory domain of the polymerase. So once the pre-initiation complex is assembled on the promoter, like we've just discussed on the previous slide, RNA polymerase II has to escape the promoter region then and begin actually making mRNA. And it's the C-terminal domain, modification of this C-terminal domain of the polymerase, which actually regulates that promoter escape. So what are the C-terminal domains, these carboxy terminal domains, like? Well, they're made up of heptapeptide repeat sequences of amino acids. And it's been shown that the number of repeats roughly correlates to the complexity of the organism involved. So, for example, RNA polymerase II from yeast, which I explained in the replication lecture, is a very simplistic, the most simplistic eukaryotic cell. CTD from RNA polymerase II in yeast has 27 of these repeats. Humans have 52 of these repeats. So it's thought that the extra number of repeats and the complexity of the C-terminal domain reflect the complexity of the organism and the need for greater regulation over transcription. So what actually happens then, once the pre-initiation complex is fully formed, that part of the TF2H transcription factor, remember this is the last one to be recruited, phosphorylates the C-terminal domain of RNA polymerase II and triggers the release of RNA polymerase II from the promoter, causing it to start making the gene. As a byproduct of this phosphorylation, in addition to RNA polymerase II starting to copy the gene information into mRNA, it also causes all these other, or most of these other general transcription factors to dissociate from RNA polymerase II and the promoter region, and other transcription factors involved in elongation then can be recruited in their place. So it's a really clever mechanism to have TF2H contain a kinase which triggers elongation and promoter escape, because obviously that's the last thing to come in. So it's another example, these things are common sense really. You don't want RNA polymerase II to arrive at the promoter and start actually copying mRNA sequence until the entire pre-initiation complex is there. So it's an example of temporal regulation again. The fact that the last thing to arrive is also the thing that kick-starts transcription means that until everything's there, transcription can't begin. So what happens once all these general transcription factors like TF2D, TF2B fall away from the promoter, once elongation begins, is you get other transcription factors being recruited, including this TF2S and this human SPT5 protein. Again, it's the C-terminal domain of RNA polymerase II, which is really important in the recruitment of these different transcription factors. And different phosphorylation patterns on the C-terminal domain promote the recruitment of different elongation factors throughout transcription. So whilst it's a kinase within TF2H which initially phosphorylates the C-terminal domain and allows RNA polymerase II to escape the promoter, it's actually a separate kinase, a second kinase, which really kickstarts transcription properly. So like in bacteria, what happens is you have abortive rounds of transcription, early elongation, where the polymerase never really gets going properly. And then suddenly transcription is kick-started and the polymerase shoots off and starts copying the mRNA at a rate of knots. That actual proper elongation, initiation of elongation, that kick-starts stimulated by phosphorylation of C-terminal domain in RNA polymerase II by a second kinase, PTEF-B. So that's illustrated here on this figure, where we can see that polymerase II escapes the promoter following phosphorylation of the C-terminal domain by TF2H, but you can see the phosphorylation is not complete, and what you've got is a polymerase kind of stutters and stalls along the first part of the gene, and then you get phosphorylation by the second kinase, PTEF-B, which really decorates the tail then with phosphate groups, hyperphosphorylates the tail if you like, and that really kickstarts the elongation of polymerase and the copying of the RNA. You can see that the phosphorylation occurs on different residues within the tail. So the initial one is on serine 5 and then the second phosphorylation event is on serine 2. Just want to say a little bit about TF2S, this transcription factor that's recruited once elongation begins. This is interesting because TF2S is also actually involved in, as well as reducing this pausing, this stalling effect which goes on early in transcription, it's also involved in the proofreading of mRNA transcripts. And it does this via activating an inherent RNase activity within RNA polymerase II. So it gives RNA polymerase II the ability to spot errors, cleave that misincorporated ribonucleotide away from the growing mRNA and replace it with the correct nucleotide. So TF2S is very important for the proofreading mechanism which exists during transcription. So you've heard from Dr. Porter last week that one of the really interesting things about the human genome is how it's organised, how the DNA sequence is organised into chromatin, and that's largely via association with histone octamers to form nucleosomes. Now, nucleosomes are a really good way of organising your DNA, a lot of information into a small space, but they present a major problem during not only replication but also transcription. So how does RNA polymerase II get around the fact that much of the DNA is wrapped around these histone octamers and is pretty much inaccessible for the polymerase? Well, this is overcome by a protein complex called FACT, so-called because it facilitates chromatin transcription. Another really imaginative name there. But it's called FACT, this protein complex. And what FACT does is it kind of operates just ahead of RNA polymerase II in the transcription machinery, and it removes chunks of the nucleosome to allow RNA polymerase II to transcribe through the gap. So it'll just remove one of the 8 histone proteins, one part of the histone octamer, to create like a hole that RNA polymerase II can then move through and continue transcribing DNA sequence. So this is explained in more detail in the molecular biology of the gene recommended textbook. Along with this lecture in general, I would definitely recommend you look at that chapter. But that in effect is how the transcription machinery gets around the problem of histone octamers and nucleosomes during gene expression. This FACT complex disassembles them, allows transcription to proceed through the gap it creates, and then it reassembles the nucleosomes afterwards. It's important that reassembly occurs because you don't want to have long-lasting changes to nucleosome and chromatin structure. So it kind of opens the thing up, transcription goes through, and after transcription machinery has gone past, it reassembles the things back together. So I'm not going to talk about this in much detail because this is going to be the content of the next lecture, mRNA processing, or the life story of mRNA, or whatever it's called in the actual timetable. But the C-terminal domain, in addition to controlling elongation and recruiting these other factors involved in proofreading, etc., fulfills another really critical role during transcription, and that's the post-transcriptional modification of mRNA. Post-transcriptional modification is a little bit of an archaic term now because recent research over the last 4 or 5 years has shown that a lot of the modifications which occur to RNA actually occur co-transcriptionally. So again, it's not the case that the cell waits until an mRNA is completely produced and then it modifies it. Literally, as the mRNA is emerging from the polymerase, it starts undergoing modification. Yeah, so it's a coupled process. But it's the C-terminal domain of RNA polymerase II that seems to interact with and bring in the other enzymes and proteins required to make these modifications. So there's several examples of modifications we'll discuss in a lot of detail in the next lecture. RNAs have a 5' cap, they have a 3' tail, and they're also spliced to remove non-coding intronic sequence. So we're not going to touch on that really more than just to mention it in this lecture. But what I am going to discuss in a bit more detail is how actually transcription stops. Because that's not a post-transcriptional modification, that's part of the transcription machinery. So how does a cell know when to stop transcribing DNA sequence? Obviously, once you've kick-started transcription at promoter, the RNA polymerase II will just continue to make mRNA until it falls off. But what tells it to fall off the DNA? Yeah. So termination of transcription is currently believed to occur via a mechanism that's been excitingly referred to as a torpedo model. So I'm assuming that a man came up with that name. It's a very phallic name, obviously, and this is, torpedoes are very phallic objects. But I don't know for certain. That's just my guess. But it's called the torpedo model, as you'll see, for a reason. So what actually happens is, following a special sequence in the actual gene, which is called a poly-A sequence, you actually get cleavage of a complete mRNA molecule then. So this figure will help to explain that. So RNA polymerase II will copy a gene until it reaches this poly-A site within the gene. And that poly-A site basically gives a signal to the cell in the emerging nascent mRNA to say, right, this is the end of the gene now. We've passed the poly-A site. That mRNA that's been produced from RNA polymerase, chop it off at that point, and that needs to then go on and get translated in the cytoplasm. Yeah. However, once you've chopped that RNA off, RNA polymerase II, meanwhile, is humming away to itself and continuing to make a second RNA. So you cleave it at the poly-A site, as you can see here in part A of this figure. But then RNA polymerase II just continues making more mRNA, and How do you tell it that, look, the gene's finished, you can go home now or whatever, you finish your job? Well, this is achieved actually via the fact that this second RNA, if you like, this kind of RNA that's been produced after the polyA site's been cleaved and the full length mRNA has gone off to become a protein, differs importantly from this RNA that's produced and then translated in the cytoplasm. The way it differs is it lacks this 5' cap. So this M7G here refers to the methylated guanine cap which exists on all polymerase II mRNAs once they're first transcribed. Importantly, after cleavage of the polyA site, this RNA that's continued to be made by RNA polymerase doesn't have that cap structure. And because it doesn't have that cap structure, it's prone to attack by a really, really active RNase, which in humans is called XRM1. In yeast, it's called RAT1, which I think is a much more evocative name, something that chews something up. But in humans, it's called XRM1. And what XRM1 does is it recognises these uncapped RNAs that are coming out of the RNA polymerase, latches onto them and starts really chewing them up, like Pac-Man. I mean, that's a really old video game reference, probably don't mean anything to you guys. But in fact, it latches onto these capless RNAs that are emerging from RNA polymerase II, starts chewing the RNA up. And it does this with such speed and ferocity that when it reaches the RNA polymerase, the idea is that it kind of torpedoes it off of the DNA. So it chews right back to the polymerase, and when it gets to the polymerase, it causes the polymerase to be displaced from the DNA, terminating transcription. That, in a nutshell, is how it works. So we've seen how transcription works with regards to assembly the preinitiation complex on the promoter, polymerase escape from the promoter and elongation, the factors involved in that, how we overcome nucleosomes and histone roadblocks to transcription, and how transcription is terminated. But what controls whether or not we get transcription anyway? Yeah, and what controls whether genes are expressed or repressed in cells? So obviously a major part of gene expression control in eukaryotes is the modification of chromatin structure. So I'm not going to talk about this because you've had a lecture on this already from Dr. Porter, but I'm sure in his lecture he mentioned some of the important complexes involved in this, such as the SWI-SNF complex, and also the acetylation, deacetylation of histones, etc. So you should look at Dr. Porter's lecture notes again in regards to how that sort of regulation affects gene expression. There is, however, a second layer of regulation that exists in eukaryotes, and this is provided by different types of DNA binding proteins, which are termed activators, insulators, and repressors. Yeah. So just via the names, you can probably already guess how they work, but we're going to go through them, obviously, so there's no doubt in your mind. So what activators do is they bind to so-called enhancer regions of the DNA. So these enhancer regions are the really distal regulatory regions I mentioned earlier on when we were looking at the core promoter. These enhancer regions can be thousands of nucleotides upstream of a promoter sequence. Yeah, so one of the big questions that have been puzzling biologists for years is how on earth do activator proteins which are bound at enhancer regions, effectively miles away from the promoter, actually affect what goes on, at that particular gene. And there are different models that have been proposed for how these might work. None of these are necessarily correct or incorrect, they're not mutually exclusive. The first example is here where you have the pink bar, which is a gene, the promoters are in purple, and then you have a long way away from those, one of these enhancer regions and an activator protein bound to it. And it's thought that perhaps the DNA can form a loop around to bring the activator in close proximity with the promoter. That's one possibility. A second possibility is that the actual enhancer and activator protein might track along the DNA sequence, so form a loop and track along the DNA sequence looking for promoters to activate. And finally, it's been proposed that there might be an accessory protein that links the two together, so that binds the activator and then starts forming a link between the activator right along the DNA until it comes across the promoter. This is an example of coupling process between proteins, similar to what we see for single-stranded diamine proteins in replication. So there's some possible models for how activators affect promoters, but in some respects, you need to take it on faith that they do. Yeah, the actual mechanism is not completely clear yet, but it is clear that these enhancer and activators are capable of upregulating the expression of genes, which are very far away from them. Now, one of the issues of that is if you've got an activator bound to an enhancer that's a long way away from the gene it affects, what's to stop it also affecting genes which might lie between the target gene and the actual enhancer sequence itself. Yeah. So what stops basically enhancers and activators just activating everything that are near it on the chromosome? Now that'd be a very loose, wishy-washy way of regulating gene expression. And we know for a fact that gene expression is very tightly regulated. So how's that achieved? So consider this diagram here. We've got an enhancer sequence here with some activator proteins bound to it, and there are two promoters for two different genes. One's distal, 5' to the enhancer, the other's 3' to the enhancer, but in theory the enhancer could affect both of those promoters and cause an upregulation in gene expression. Yeah, so it could affect expression of the promoter there, or it could affect expression of the promoter there. Now, insulators exist to make sure that that doesn't happen, or there's a way of making sure that doesn't happen if the cell deems it necessary. So what insulator sequences do is bind to these insulator proteins and cause the enhancer to be unable to exert its effect on the promoter in one direction, but retain the effect in a different direction. So as you can imagine, if you start getting very complicated combinations of enhancers and insulators across a DNA sequence, you can set up very complex regulatory networks with regards to gene expression. So what are these enhancers and activators and insulator proteins? Well, effectively they're all obviously DNA binding proteins. By their very nature, they need to be able to bind DNA. Important fact about most of these DNA binding proteins and DNA binding proteins in general is they tend to bind as homodimers. Yeah, so they're binding pairs. Eukaryotic DNA binding proteins can be a little bit more interesting and bulk that rule and bind as what's called a heterodimer. So you can have two different DNA binding proteins that bind together as a heterodimer, rather than just two proteins coming together to bind the DNA as a homodimer. And each of these DNA binding proteins tends to include some of the classical DNA binding domains, which you may have encountered in other lectures. Again, Dr. Porter may have mentioned these. Some examples of classical DNA binding domains include homeodomains, zinc finger domains, leucine zipper domains, helix turn helix domains. So these are all examples of domains which, if found in a protein, indicate that that protein is likely to be a DNA binding protein. So the leucine zipper, for example, is shown here. So this is a structural image of what AP1, which is a transcription factor, looks like. It's also known as phosgene, and how it interacts with the DNA. And what you have are these two alpha helices that kind of form like a zip structure. And where the zip's opening at the bottom in this homodimer, that's the part that interacts with the major grooves of the DNA to bind to the DNA molecule. So how do the activators actually work? So I've said that they enhance expression, but how do they do that? It's not by magic. So they effectively activate gene expression by indirectly recruiting RNA polymerase II to the gene. Yeah, so they don't directly recruit the polymerase, but they have the function to indirectly recruit the polymerase to the gene. And in the simplest term, this can be achieved by an interaction with a subunit of the preinitiation complex, such as TF2D, or it can be achieved by an interaction with this complex I mentioned at the beginning of the lecture, the mediator complex. So the mediator complex is a really fantastically complicated multi-protein structure. It contains many different proteins. And it's known that the activator proteins, like we see in purple here at the top of this figure, are able to interact with this mediator complex. And then the mediator complex itself interacts with the general transcription factors and RNA polymerase II to help promote formation of the preinitiation complex of promoter sequences. Activators can also affect nucleosome modification enzymes such as SWI/SNF. Again, I'm not going to mention that because it's going to be repeating what Dr. Porter told you, but they're able to manipulate these histone modification enzymes to positively affect gene expression. So the mediator complex, then, to come back to that briefly, is it's believed to be involved at some level in the expression of all class 2 RNAs, in other words, all mRNAs in eukaryotic cells. So it's an incredibly important complex, the mediator complex. And it's fantastically complicated. In humans, it's 1.2 megadaltons in size, so it's massive, it's got 26 subunits. And considering its importance, it's relatively poorly understood. But what's appreciated is that such complexity obviously offers a really dizzying level of potential regulation to gene expression. So having taken into consideration all the other things we've already said about how expression is regulated, the C-terminal domain of RNA polymerase II, what we then throw into the mix is this massive multi-protein complex which interacts with all these really distal enhancer elements and different activator proteins to add yet another layer of complexity to gene expression. But if you think about it, as humans, we need that level of complexity to be there. One of the big questions is why on earth we've got the same number of genes as a worm. The Human Genome Project's revealed that. We'll touch upon that again next week when we talk about mRNA processing. But it's not just the number of genes, it's not just the size of the genome that's important. Size isn't everything. It's how clever you use a genome, how you're able to use the information. And as humans, thanks to the mediator complex and many of the other eukaryotic protein complexes which aren't present in bacteria, we get a lot more bang for our buck from our genomic information than some of these simpler organisms do. So there are other mechanisms as well which you need to consider. Repressors I've not talked about yet. I mean, obviously the clue's in the name. These things repress gene expression, turn it off. There's several mechanisms which they can use to do that. They can actually compete with these activators at these enhancer sites, so that they'll bind the enhancer site and prevent an activator from binding there, thus in effect down-regulating gene expression. They can bind next to where an activator is bound, an enhancer. So bind at a separate domain and then interact with the actual activator protein itself to inactivate it. And they can also interact directly and inactivate general transcription factors. So there are some repressors which will bind directly to TF2D or Tata binding protein in order to repress expression by, again, making those proteins inaccessible to the activator proteins which would normally interact with them in order to bring in RNA polymerase II. So there are several ways in which repressor proteins can function. Epigenetic control is another important aspect of DNA expression. Again, Dr. Port will have touched on that, so I'm not going to concentrate too much on it. You should have a look into a bit, if you want to do some extra reading that's interesting, have a look at the ideas that there might be a histone code. So modification of histone proteins adds yet another complex layer of regulation to this entire process. Methylation, epigenetic control is incredibly important in gene expression and also in development. As we'll find out later in the module, certain general transcription factors have now been used to reprogram cells, so to turn one cell type into a different cell type for therapeutic benefit. And we'll talk about that later in the module when we do an induced pluripotent stem cell lecture. So I mentioned at the beginning that there are 3 polymerases in eukaryotes. So just to finish with, let's mention what the other 2 of those are. The first of those is RNase polymerase 1, and this is RNA polymerase is responsible for transcribing just one gene. However, it's a very important gene. It's a ribosomal RNA precursor. So this is the RNA which forms part of the ribosome. So this RNA is by far the most highly expressed RNA in the entire eukaryotic cell. Because every single ribosome in your cell which makes protein requires this RNA in it to work. So that's one potential reason why it's got its own dedicated RNA polymerase. It's quite a simple setup. It just needs 2 general transcription factors. SL1, part of which is the Tata binding protein. So Tata binding protein is found in this SL1 complex. And another general transcription factor called UBF. So that's RNA polymerase 1, and that's transcription of the ribosomal RNA precursor. And then the other one's RNA polymerase 3, and this kind of does a few other different types of specialized RNA. So 5S ribosomal RNA, which is another ribosome RNA, is transcribed by Pol3. tRNAs are transcribed by Pol3. Small nuclear RNAs, which are important components of the spliceosome, as we'll hear next week, are transcribed by Pol3. And some microRNAs, we'll touch on microRNAs when we talk about regulation of translation. They're also transcribed by RNA polymerase 3. With regards to accessory factors, you need TF3A for the 5S RNA and TF3B and C for the tRNAs. So again, it can't just do this job on its own, the polymerase. It requires these accessory factors, these general transcription factors to function. I'm not going to say any more about those. As I said, the most important polymerase is RNA polymerase 2 from your point of view, but you need to understand that these other polymerases exist and you need to know what they actually do. So, to finish with, let's have a think about transcription in human disease. One good example of how when errors in transcription occur can lead to disease is SCAR17, which is an ataxia disease, spinocerebral ataxia 17, and also Alzheimer's disease. And they've both been linked to a specific type of mutation in Tata binding protein. It's one of these triplet expansion mutations, again, like we see in Huntington's disease. In this case, it's a CAG expansion. And it's a causative for SCAR17, and it's been associated with Alzheimer's disease as well. In SCAR17, it's now known that the expansion actually interferes with tata-binding protein's ability to interact with TF2B. Yeah. So if you remember, TF2B comes in and stabilizes the entire preinitiation complex. When you get expansion of this CAG domain, that causes that process to be perturbed, so you can't get stabilization, and it leads to SCAR17 as a disease. The Alzheimer's link's a little less clear, but it's thought that the expansions cause tata-binding protein to become insoluble. Yeah, so it forms what are called inclusion bodies, insoluble inclusion bodies in the cells, and that's been linked to the formation of Alzheimer's in certain neuronal cells, etc. So the link's not as clear, but there's definitely a link there between expansion in tata-binding protein and the development of Alzheimer's disease. Another interesting example is cocaine syndrome, and also that kind of comes under the umbrella of this disorder called xeroderma pigmentosa. So one of the things which transcription also does, in addition to make mRNA, is it's kind of an important part of the DNA repair mechanism. So both of these diseases actually arise due to errors in these DNA repair proteins. So for cocaine syndrome, it's specifically ERCC6 or ERCC8. Mutations in these genes lead to cocaines. Xeroderma pigmentosa is a little more complex, and it's an example of genetic heterogeneity, that one disease can be actually caused by mutations in several different genes. And the reason why different genes can give you the same disease is that the disease arises due to a loss of a particular pathway, rather than just one gene itself. And obviously all the genes which have mutated give rise to xeroderma pigmentosa function within that pathway, in that same pathway. So in both cases, if you lose these DNA repair mechanisms that exist and that are coupled to transcription, the symptoms include extreme photosensitivity. So obviously UV damage to the skin is a major source of DNA damage. We talked about that in molecular genetics. And if you lose the ability to repair that, then you become very photosensitive. Greatly increased risk of skin cancer, malignant melanoma, etc. These disorders are very rare again, 1 in 200,000, but again, like we saw for Werner's syndrome, there's an increased risk in the Japanese population, where it's about 1 in 40,000, the frequency of these disorders. So on the right here, this figure, it's kind of showing how these things arise. You've got, in addition to the transcription factors you recognise, TF2H, etc., these accessory proteins which bind to it and polymerase 2 during transcription and monitor for DNA damage. If they come across a piece of DNA that's damaged, they'll repair it before the gene's made. Yeah? In these diseases, some of these accessory factors, these DNA damage proteins, are mutated and lost. By wiping one out, you more or less wipe out the function of the entire process. So this is an example of somebody who's unfortunate enough to have xeroderma pigmentosa. You can see multiple large malignant melanoma on the face, very freckled expression. So it's a really, nasty disorder to suffer from. So just to summarise quickly then, eukaryotes contain 3 polymerases. We've talked about what they all do. The preinitiation complex forms on these eukaryotic promoters, and it forms in a set order. Yeah? Much of the order is about stabilising previous complexes which have already bound. Remember, tata-binding protein bends the DNA almost by 90 degrees due to its almost unique beta sheet minor groove interaction that it forms with the DNA. Once you've got recruitment of the preinitiation complex, elongation is kicked off by phosphorylation of the C-terminal domain of RNA pol 2. Different factors, including TF2S, which is involved in proofreading, are then recruited to the RNA polymerase complex, which is really kick-started by phosphorylation by PTFB. Termination is retrieved via XRM1 and this torpedo mechanism, which works by recognising RNA which doesn't have a 5' cap. And fine control of transcription is achieved via these activators, repressors and insulator proteins, these DNA binding proteins, which can bind very far away from the promoter but still exert an effect on expression of a gene. And as I've said just now, obviously errors in the transcription machinery associated with a number of human diseases, including xeroderma pigmentosa and Alzheimer's disease, cerebral ataxia as well. So again, there's an excellent couple of chapters of this about transcription in molecular biology of the gene. It's on the recommended reading list for the module, so you should have a look at those chapters.